Firstly, I would like to thank Wonka asked me to give a presentation about root and root soil interaction in intercrop system. We have did a lot of research on that aspect in the past more than 30 years. So today I'd like to give three parts of root-root interaction and two parts of root-soil interaction. That included the root-root interaction and the competition recovery principle. Second one is the root-root interaction and the efficient use of phosphorus. Third one is the root-root interaction and efficient utilization of nitrogen. And root soil interaction and soil fertility, and root soil interaction and soil multifunctionality. Finally, I give some conclusion. First, I would like give some root interaction and competition recovery principle. As you could see anyway, see the difference between the three rounds of maize. Can you see any difference between three rounds of maize? The maize is the same cultivar, sow same time and fertilizer same time, same amount. Also, what's the difference is neighbor. So that rounds of this rounds of maize neighbored with wheat, and the other rows of maize neighbored with faba bean. You can see these rounds of maize grow very healthy, very good condition. So we defined that is interspecific competition between wheat and maize. Interspecific facilitation between maize and faba bean, both crop grow very well. In this system, maize was suppressed by wheat, but wheat over yielding in the competition. Okay, that is a typical ecology principles for competition and interspecific facilitation. So first, I would like to show some competition recovery principle in rally intercropping. That was founded by our research group very earlier in 1990s. That is published in 2001 in Field Crop Research. So what's the competition recovery principles? That means that in the rally intercrop system, the wheat, the maize, or soybean has some core growth stage. There are competition during the core growth stage, and after wheat harvest, there are some recovery after for maize. You can see that this is a core growth stage, above ground biomass. You can see compared to salt wheat, intercrop wheat over year greatly compared to this one. However, for maize, the before wheat harvest here, the intercropping maize was suppressed by wheat because compared to store maize, the biomass of maize, intercropping maize was decreased compared to store maize. However, after the wheat harvest, intercrop maize has rapidly grown. So finally, it's also get more or equals to yields of the store maize. So that's a competition and the core growth stage and recovery after early maturity crop harvest. So we would like to know what's the mechanism underground. So we dig a very big soil profile to look at the root distribution in the intercrop system. You can see this is a monocultured maize from the base of the maize plant. The roots can distribute separately, laterally about 40 centimeters in the monocultured maize. And in the intercropped maize, here is wheat. You can see the root distribution laterally only about 20 centimeters. So we quantify the root distribution like this. We published that in 2006 in Oil Ecology. So if you interested in that, you can read the paper. So that means the roots of the intercrop wheat extended into the underground space occupied by maize, whereas the roots of intercropping maize did not extended into the underground space occupied by wheat. So we would like to know what's the mechanism behind the roots avoidance of maize. So firstly, we look at the root resources competition because based on general ecology principle, competition usually derived from the resources. So firstly, we look at the resources competition. In the field, we applied different rates of nitrogen from 50 to 350. That's a really high nitrogen application. So you can see the roots of maize intercropped with wheat only just occupied themselves space underground. Very high nitrogen application do not distinguish the phenomenon. So nutrient competition accounts are not completely the phenomena of the roots of maize treated to avoidance to wheat roots. So we hypothesize that root exudative would play a role in the root avoidance. We published that in 2015 from the soil. So we, in the greenhouse, we conduct a pot experiment, use this kind of device to, this is a control just for maize grow without any neighbor and root exudative from wheat. So this is neighbored by wheat. Wheat was grow for a week. After that, maize removed to the grow together. Also, wheat root just grow in this container for 1 week. After that, wheat roots, wheat plant was removed from the container. Just has some root exudative of wheat keep in a container. So we move the maize plants into the container. So we found that means wheat roots exudating and wheat plants and control. We found that the root length of maize was reduced significantly by 37% when neighbored by wheat seedling. And by 40, will grow with substrate enriched with wheat root exudative, compared with maize grow along under sterile condition. That means root exudatives play a very important role in maize roots inhibition. So we use another experiment like this. There are some control, also collect some root exudative from wheat, barley, and maize, added to this device cultivated maize. So we look at the roots, total roots length of maize. We found the maize roots treated with wheat roots. Sorry. Wheat roots. This is had a 64% of short root length than maize roots was only treated with nutrient solution. So maize or barley's roots today did not affect the maize root density. So we want to know what component of inner root exudative have this function. So we selected three lots of chemicals. We found the MBOA, very complex chemical substance, a kind of alleloplasty chemicals in which root exudative forced the roots avoidance by inhibit roots growth of maize. So you can see we use this device to collect root exudation in monocultured wheat, monocultured maize, and mixture of wheat and maize. We found the concentration of more MBOA concentration in the mixture is significantly higher than that of maize grow alone, and maize grow alone, and the weighted means of wheat and maize grow alone. So that means when the two crops grow together, wheat has a much more exudative MBOS exudative. So we also found that MBOA inhibited maize root growth, but not had no effect on wheat roots under the same concentration of MBOA. You can see this is the MBOA standard concentration. If there's a 0.3 milligram per liter, the total root length of maize was decreased. But if the wheat roots do not have any response to different levels of MBOS concentration. So in the same media, the wheat is not sensitive to MBOA, but maize is very sensitive to MBOA. So in the intercrop system, wheat roots exudated inhibited the roots growth of intercropped maize. So that's we explained the what's that interspecific competition and recovery. So that's just a competition. So we like to show why it is a recovery after wheat harvest. We have a lot of field work at around the 30 September, that is almost maturity of the maize. You can see the roots in the soil profile. For the roots in the soil maize, the very little roots. But if the maize intercropped with the wheat and the faba bean, there are lots of roots still kept in the soil profile. So the roots of intercropping maize has a longer lifespan than those of soil maize. Secondly, we also measured the nitrogen uptake by maize for roots length after wheat harvest. We found that this is intercropping maize, the monocultured maize. The nitrogen uptake efficiency, that means nitrogen uptake rate per root length after wheat harvest was higher than soil maize. So the recovery of intercropped maize was attributed to the longer root lifespan as well as nutrient uptake efficiency of intercropped maize. So that's why in the wheat and maize, wheat and soybean intercrop system, there are competition and recovery pretty simple. Next, I will show you some root-root interaction and efficient use of phosphorus. Usually in a field condition, we use root barrier to determine the contribution of below ground interaction to nutrient uptake and as well as yield advantage. I will not show yield advantage here, just for phosphorus uptake. The root barrier like this, the insert between two species. The barrier can be a plastic sheet, that means no root interaction, and a nylon mesh between two species. There are some interspecies rhizosphere effect because the nylon mesh do not permit the root go through other space of other neighbors. So that means that the roots just limited in themselves on the ground space. So also there's some no barrier. If there's no barrier, there is an interspecies rhizosphere effect and as well as a special effect. So from this experiment, we found that interspecies root interaction enhance phosphorus uptake by maize, do not influence phosphorus up by faba bean, and finally increase the phosphorus uptake by the intercrop system. So we would like to know why, what's the mechanism behind the interspecific facilitation of phosphorus uptake. So we, in a greenhouse, we conducted pot experiment to look at the interspecific facilitation of phosphorus. We use this is no any phosphorus added some iron phosphorus, added some calcium phosphorus, aluminum phosphorus. This is a soluble phosphorus. So also we use a mesh barrier, no barrier, and a solid barrier. You can see there is no any difference between not any difference in faba bean spimers between different treatment. However, you can see the growth of maize has a different performance in a different treatment. If there's a no solid barrier, that means no any rotator reaction between two species. You can see the maize grow much less compared to no barrier, the mesh barrier. Similar is for alumina phosphorus and calcium phosphorus and no phosphorus addition. So we look at the phosphorus uptake by two species. You can see root-root interreactions significant increase phosphorus uptake by mass or average for all resources of phosphorus sources compared to uptake without root-root interaction. This is a non-rotator reaction, rotator reaction, or rotator reaction. Phosphorus by mass is a rhizosphere connection, that is a mesh barrier. Midway was 30 grades that without rotator reactions, solid barrier without any phosphorus addition. 160% greater with iron phosphorus addition. So 56% greater with this one. So there was no any significant difference in phosphorus uptake by Faba-B between the mesh barrier and the solid barrier. As well as there also there are different there is no difference between phosphorus sources. That means Faba-B can use various phosphorus forms in soil, but maize cannot do that. Also, if there are some rotator reaction between Faba-B and the maize, the phosphorus uptake by maize increased. So we would like to know what's happening between two species. In the laboratory, we found Faba-B acidify its rhizosphere while protein exudation from roots, whereas maize cannot do that because Faba-B yellow one means pH decrease. So in Faba-B's rhizosphere, the pH decreased. But maize do not do that. If the pH in the rhizosphere decrease, that will be mobilize insoluble phosphorus in soil. So, for example, for alumina phosphorus and some iron phosphorus can be dissolved by exudative. So that we explain it why Faba-B can use different phosphorus forms. And we also measured the organic acids in rhizosphere soil. We found that concentration in rhizosphere soil in Faba-B is for malate in Faba-B for nitrate it's much higher than soil-B maize. So that's also another mechanism for phosphorus mobilization for Faba-B. So we for the phosphorus facilitation between phosphorus mobilized species and no phosphorus mobilization species in intercropping. The phosphorus mobilized species can utilize the spirally soluble phosphorus, which result from greater exudation of organic acids and greater rhizosphere acidification. So it has the phosphorus acquisition of no phosphorus mobilized species were derived from root-root reaction from neighboring phosphorus mobilized species, which mobilize insoluble phosphorus from soil. That's for phosphorus. Also, we found enough under field condition. Intercropping has the apparent recovery of fertilizer phosphorus by average 20 percentage points compared to monoculture at the 12th year and 30 years that published in this journal. If you're interested, you can look at the details in the paper. So next I will show some root-root interaction and efficient utilization of nitrogen. We still use this root-berry experiment under field condition. We found that root-root interaction between two crops increased the nitrogen acquisition by two species in the flower bean maize. You can see the no-root interaction, partial root interaction, and the completely root interaction increase the nitrogen acquisition compared to the no-root interaction. Well, we'd like to know what's happening below ground. Actually, in our really earlier research in 1990s, we found the why mechanism on behind of the nitrogen efficient use in legumes cereals intercrop system. That means the cereals compete much more soil nitrogen, mineral nitrogen, which alleviate the soil nitrogen inhibitory effect or nitrogen fixation of legumes, and leads to overyielding of cereals. So that is published in cereals papers in different lab, especially in plot soil. I will not go to the detail about that. So actually, in our past research, we do not find any, do not find the competition cannot explain it every intercrop system for nitrogen, enhance the nitrogen fixation in legume and cereals intercrop system. So we have another hypothesis. Probably root-root reaction plays some important role. So we conduct this kind of collect root use data for maize, barley, and wheat added to this equipment. So we this device, there are some 40 sets, aluminum foils outside, and a nutrition solution with 0.1 millimeter and 2 millimeter, 2 millimole. So like this device. So we found the different effects of different root udatities or nodulation. We found only 8 root udaty of maize facilitated nodulation of faba bean. You can see red bar. That means maize root udaty compared to that. So we found some root udaty signals in root mutations. There are some, for example, for geneticity. That is a very important signal for talk between rhizobia and legumes. So we also measured some genes relative to nitrogen fixation and as well as natural fixation enzyme. So you can see the treatment with Faba-based maize root utilities, the relative genes expressions in the root utilities of maize was increased compared to without maize root utilities. So genicity, a kind of flavonoids, may act as a key signaling promoted nodulation of Intercropping Faba-B by maize, but not by wheat at all, barley. So for another sector mechanism behind of the enhanced nitrogen fixation of intercropping legumes with maize is that maize promoted nitrogen fixation. Maize root exudative contain significant flavonoids and promoted flavonoid synthesis in Faba-B. Therefore, triggering modulation with the involved of the hormone signaling and stimulated the nitrogen fixation while enhanced activity of proteins involved in nitrogen fixation and the genes expressed and the physical level. Okay, I will go to the root soil interaction now for soil fertility. So our question is, what's happen in the real world on the field condition? Because in the laboratory, we found the root-root interaction play a very important role in phosphorus uptake, nitrogen efficient use. So how complex root-root interaction influence agricultural sustainability? We conduct a field experiment in the northwest part of China, as I showed in the day before yesterday. The very dry area, I think Wolfgang was visited over there. Lots of long-term field experiment we conduct in different oases that has different fertility levels. For example, here is a very young oasis, just reclaimed 3 years. We conduct a field experiment here. That is experiment 4. And experiment 3 in here is reclaimed 20 years. And as well as a very old oasis, that is very fertile soil. We have 2 experiments. Here, there are some very fertile soil. Soil organic matter is almost 20. Here is about 60, and here only 6 rounds. So that is a great variation in soil fertility. So we conduct, this is our 4 long-term field experiments in irrigated area in northwest part of China. First way is established in 2003 in fertile soil. There are 3 monocultures of wheat, maize, and Faba-B, 4 crop rotation, and 6 intercropping, and there are 3 replicated, totally 39 plots. And the second experiment and the third one located in second one is the same as site, and the second one is located in other oases. So there are 3 phosphorus application, 5 crop monoculture, 4 intercropping, 3 replicates on 2 sites. 81 plots each site. So third one is a very poor soil, also start in 2009. two rhizosphere treatment, the 5 nitrogen application, 3 crop system, 2 monoculture of faba bean and maize, faba bean maize intercropping to replicate totally 90 plots. So we found the use the data in up to 2018. There are for first experiment already 60 years, and the third one, second one, third one, and the fourth one is about 10 years. So totally about 60 or 46 years. We found there's a significant over-yield from intercrop compared to monoculture. You can see this is blue one is the intercropping, red one is monocultured. Average gray yields across all 4 experiments and all 6 crop combination was about a 22% of greater in intercropping system than in monoculture of the same crops. Across all 4 experiments and all crop combination, intercropping yields increased by an average 2.52% per year. This is intercropping, but monoculture of the yield of the same crops increased by only 1.67% per year. This result showed that yield benefit of intercropping was not just sustainable, but also increasing. That's for. We also measured the temporal stability of yield. Use this temporal stability is about average yield divided by STDE, more years. So we got this in intercropping and monoculture. On average, the year-to-year temporal stability of intercropping yields was significantly higher than that of monoculture for both the equal fertilization and the optimal fertilization treatment in all 4 field experiments. Intercropping can stabilize the gray yield over time, especially compared to monoculture. We also measure the soil fertility parameters. We firstly the abundance of large soil aggregate. It's the 4th to 6th year after experiment conducted. So for equal fertilization and optimal fertilization, both intercropping, the larger soil aggregate was significantly higher than monoculture under both fertilization conditions. We also, under the 4th to 6th year after experiment conduct, we found some for the equal fertilization experiment relative to crop monoculture, intercropping increased total nitrogen and as well as soil organic matter and very poor soil. But soil organic matter, total nitrogen in soil and the pH did not change significantly and fertile soil. So that is at the beginning, first of 6 years. However, we in the later and the 30 year, we still to measure the soil total carbon and the soil organic carbon as well as the soil inorganic carbon. After 30 years of continuous intercropping, soil total carbon and organic carbon concentration show significant increase, 2.2 and 5.8% compared to monoculture extent. This is last year just published in this journal. If you're interested in that, you can go to details. We also found intercropping enhance not only soil organic carbon, but also in soil inorganic carbon in sandy soil and humus and 30th years. Also, that is also published last year. You can, I do not go to the details. Also, we compared the effect of short-term and long-term intercropping or soil total nitrogen. You can see the red way the long term and blue way the short term. You can see there are some after 12 years of intercropping in 2020. Intercrop significantly increased the soil total nitrogen in legumes maize intercrop system with phosphorus application. Intercrop significantly increased the soil nitrogen, total nitrogen concentration in non-legumes maize intercropping without phosphorus application. So, okay, I think okay, probably finally.
It's totally okay because Marie Svetlov cannot come today.
Oh, cannot come. Okay.
You can spend a next.
So I can. I would like to show some root soil interactions and soil multifunctionality, because in our previous study just focus on soil carbon and nitrogen, do not pay more attention other soil parameters. So recently we focus on the soil multifunctionality. So what's the soil multifunctionality? That is a very important part of ecosystem multifunctionality. Refers to the capacity of soil to maintain multi-ecosystem function and service. So how does intercropping influence soil multifunctionality? So still we use a long-term field experiment in the Jintai site. That is medium soil fertility at the beginning. The long-term field was initially in 2009 in northwest part of China. And the main plant vector was a 3-phosphorus fertilizer application, 0, 14, and 80 kilogram phosphorus per hectare. The sub-plant vector was a 9-crop system, included a 5-crop monoculture of chickpea, faba bean, oilseed rape, soybean, maize, and a 4 intercropping system with chickpea maize, that one, faba bean maize, and oilseed rape, maize, and soybean maize. We divide the soil multifunctionality into 6 classifications, including the physical structure, carbon, nitrogen, phosphorus cycling, and other elements cycling, and abiotic stress regulation. It's about for soil structure. We look at the bulk density, soil moisture, soil compaction, and mean weighted diameter of different class aggregates. And for carbon cycling, we focus on the soil organic carbon, micro biomass carbon, and different enzymes. Although also for nitrogen cycling, there are total nitrogen, micro biomass nitrogen, and nitrogen cycling relative to the enzyme in the soil. Also, phosphorus, total phosphorus, available phosphorus, and the micro bimore phosphorus and phosphates included acid phosphates and alkyl phosphates and other parameters. So firstly, we look at the soil multifunctionality under intercropping and monoculture. Soil samples were collected in the 30th year. That means in 2021. So firstly, we look at the soil multifunctionality under intercropping was 20% higher than that in monoculture across all crop combination and phosphorus application. Among crop combination, intercropping show consistently positive effect on the soil multifunctionality compared to that of the weighted mean of monoculture.
No, maybe I missed it, but did you explain how you calculate this index for soil multifunctionality?
Yeah, average all
Just
all
out.
parameter. Yeah,
Okay.
we use a different method.
Okay.
So I show that later.
Okay. Okay.
So this just for intercropping compared to monoculture and different combination intercropping monoculture and three phosphorus application intercropping monoculture. So that is a whole picture for soil multifunctionality. Phosphorus it has the soil multiple functionality, especially under monoculture. So yeah, we use a different method to calculate the soil multifunctionality. Use a linear scoring edit or no linear edit. We also use a minimum data set and a total data set. Total data set means more than 30 parameters. We use the minimum data set is just for very important one. So the linear, whatever we use any method, we found that the soil multifunctionality was higher in intercropping than that in monoculture by both linear and non-linear scarcity additive method. There was a significant positive relationship between soil multiple functionality calculated by average approach of total data set and the minimum data set. That means we can use a minimum database to examine the soil multifunctionality. This paper just published online yesterday. I think that's you can look at that. Soil multifunction over time. We use a minimum database to calculate the soil multifunctionality over time from 2012 and 2024. You can see we use two methods, linear scoring and non-linear scoring. Generally, the soil multifunctionality was increased over time. However, the intercropping increased more quickly than monoculture. So also we measured some function genes relative to carbon, nitrogen, and phosphorus cycling. So we found that intercropping significant increase the relative abundance of genes relative to carbon. This was relative to carbon intercropping monocultured and relative to nitrogen in intercropping and monocultured, phosphorus cycling in intercropping monocultured, and sulfur cycling has no significant difference between monoculture and intercropping. The abundance of the functionality genes relative to carbon, nitrogen, and phosphorus cycling was correlated significant with the individual function of soil physical structure stability, carbon and nitrogen cycling, and furthermore relative to soil multiple functionality. We also found that soil multiple functionality was associated with significant the abundance of function bacteria genes relative carbon and nitrogen cycling. For example, there was a significant positive correlation between soil multiple functionality and the relative abundance of functional bacteria relative to aromatic compounds, degradation and urinalysis, nitrate, respiration, and denitrification. So there was a significant negative correlation between the soil multifunctionality as well as the individual soil function, for example, for physical structure, stability, and nitrogen, carbon cycling, and abiotic stress. That means there is some synthesis or some trade-off between individual functions. I think the soil multifunctionality and the individual functions was closely associated with the relative abundance of phyte separate, that is a kind of fungi. Also, the crop was a significant and a positive and correlated with the individual function. That is the individual function, and this one is the gray yield was positive correlated with soil multifunctionality. So intercropping increased the soil multiple functionality, therefore further increase the gray yield. Okay, finally, I'd like to give some key messages. Compared to monoculture, intercropping can enhance the soil fertility provider up to 22 more food per unit land. The intercropping can help increase the year-to-year reliability of crop production in some cases. Root-root interaction can explain the competition recovery principle via root avoidance, driven by root eudatives and enhanced root life span and uptake efficiency of roots. Root-root interaction benefit to efficient use of nitrogen, while by enhanced nitrogen fixation and efficient phosphorus use by interspecies facilitation or phosphorus driven by root eudate of phosphorus mobilized species. Soil interaction enhances soil fertility as well as multifunctionality by shift of soil microbial composition driven by different crops. the relative abundance of functional genes, relative carbon, nitrogen, phosphorus cycling increased in intercropping, and this produce a positive effect on soil indicators, therefore increase soil multifunctionality. Okay, I would like to say thanks all my collaborators, colleagues, and postdoc, master PhD student in past decades. Also, some for the National Science Foundation of China that is have us financial support. Thanks.